Hope you had time to study and answer your bell work question today for part 3 of our notes. So let's move on. So we moved on from fossils. That was the first evidence for natural selection. So we're going to move on to patterns of development, and that's called embryology. And so once you fill your notes out, we're on slide 56. Take a look at that right there and tell me what you think. Well, think of what you think. So if we can compare the early stages of development among different organisms, we see that they have similar structures, similar features. For example, the gill slits. All of these embryonic stages of animal look very similar. And this is an actual human embryo that's 4 weeks old, right there. And you can see the tail and these gill slits here that turn into your trachea and your voice box. And it's quite amazing. So by looking at this design, we can see evidence of a common ancestor. The third aspect is biogeography. And again, if you're needing to know where we are, we're on page 9 of the notes, towards the top there. So this is the geographic distribution of species. Biogeography. Bio meaning life, geography meaning where on the earth, okay? So kiwis, look at that little bird. They're so odd, but they're so cute. They live in New Zealand, which is a big island nation off of Australia. Cassowaries also are from down in that region. These are babies. Ostriches from Africa. You can go to a live website in the Namib Desert, live feed on YouTube, and you can see these guys in their natural environment, running around, okay? And emus. Emus also from Australia. So, and the cassowaries from, so these islands, these have these big birds. These things they have these claws, they will tear you up. It's crazy, but anyway. So Darwin noted that these organisms had similarities in physical appearance. And same thing when he went to the Galapagos Islands. Resembled species of South American mainland, so they knew there must be some relationship between them. Here are some more. We've got in North America, we've got the American beaver. In South America, we have the coipu, the muskrat in North and Central America, the capybara, the largest rodent, and here's the beaver picture. Similar characteristics are teeth, limbs, hair, nose, diet, and habitat. So that means they probably have a common ancestor, right? Settled in different regions, and then mild changes in their surroundings made that species adapt. So after a long time, 4 different species evolved that came from one ancestor. That's called speciation. The fourth one is comparative anatomy, and we talked a little bit about this in the whale notes from the last set of notes, last week, where you have very similar radius and ulna, the forearm bones, the humerus, the carpals, that's your wrist bones, and the phalanges, your fingers, finger bones. These are called homologous structures. Remember, homo means the same. Different functions. We don't walk on our hands, but cats do, and dogs and other quadrupedal. We're bipedal. Quadrupeds, like horses and cats. And then whales use theirs for swimming, and bats use theirs for flying. All mammals. And you can see the fingers, a term should be used loosely, in an elephant seal, but it's a flipper. Common ancestry. So homologous structures show divergent evolution. So you have, over time, you have a parent species that has speciated, diverged, like Darwin's finches. You have the initial bird who branches off into other species. Divergent evolution. Starts with a common ancestor. And then the accumulation of differences and something called isolation from the other one, so there's no interbreeding, would create a new species eventually. Speciation. Now, similar but different, okay, is analogous structures. So these structures are, they have features that have different structure but similar function. The other one was the same structure but different function. This one's similar structure, same function. All right? So, and they evolved in response to similar environmental conditions or pressures. The pressure here was, I need to swim faster. So the organism that had the best adaptation for that would be passing on their genes to their offspring. So sharks have a flipper, but look at the difference in structure, okay, between a fish, a bird, and a mammal. But they all do the same thing. They propel the organism through the water. Look at the flight mechanisms of a moth. That looks like a pterodactyl, I guess. A bird and a bat, okay? Here you've got feathers in a bird, chitin in insect. Any insect wing is made out of that protein called chitin. Skin over a bat or a pterodactyl. And then you've got cartilage versus bones as well. So you've got mammal, bird, reptile, insect. And analogous, same function, different structure. You're going to have to test yourself on those two words, homologous and analogous, because you know they're going to be on the test, and you know you're going to have to know the difference. And really the best way to do that is with flashcards. So if you're not making your flashcards, and you're not doing good on tests, you need to do that. Otherwise, you're going to keep doing bad on tests with questions like these two things that are similar in sounding and similar in meaning. Okay, so analogous structures show convergent evolution, convergent rather than divergent. No recent common ancestor. Convergent evolution is when organisms not closely related independently evolve similar traits as a result of living in similar environments. So again, you've got streamlined body, and fins to stabilize their movement in the water. But these organisms are not related, closely related at all. Mammals, you got sea lions, whales, they're mammals, but they're more closely related. But then you got the sharks, which are obviously not. Sharks have been around a lot longer than mammals. Sharks have been around since the time of dinosaurs. Vestigial structures. So we've moved on from A, B, and C in your notes to vestiges, or A and B. Now we're at C. And these are structures that are no longer used by an organism, that they're still present. We talked about this last time when we talked about the leftover hip bones in whales and dolphins today. That's an example. So here's some more examples. The appendix in humans, mine had to be removed. I have a nice little scar about that long right here, about 23 years ago, 24 years ago. And I, perfectly fine, look at me. I don't need an appendix. Why? Because it's a vestige, left over. And sometimes this can get infected, inflamed, and have to be removed. That's called appendicitis. And then you get an appendectomy. Anything with the word ectomy at the end means removal. And they believe that our ancestors used that extra piece of intestine, it's about the size of your pinky, right there, to digest stubborn plant tissues. Like our ancestors ate more plants off of like trees and stuff like that. This is called erector pili. You ever seen a cat fur puff up because it's scared or angry? And that's the same reason we get goosebumps. Not the reason, but it's the same mechanism of goosebumps on our skin to make us appear more large and threatening, but we don't need that anymore. Tailbone for tails, and tails are for balance. We don't, if you removed that, it wouldn't affect us at all. And then the third eyelid, the nictitating membrane. A lot of organisms have a nictitating membrane, sharks, toads, many things that bite their prey with their mouth. Because that third eyelid, you have an upper eyelid and a lower eyelid, upper and lower, and then you have, in other animals, these animals, they have this third eyelid that pretty much covers their entire eye whenever they go to bite something. Because whatever they're biting is probably going to try to fight back, right? So an owl trying to bite a rabbit, the rabbit's got teeth and nails and scratches, and an owl losing an eye would not be good for the owl. Same with sharks, toads, crocodiles, and cats. So they have that nictitating membrane. We have a leftover called a semilunar fold right there in the corner of our eye, and that is a vestigial feature left over. We don't need that. So other organisms, we talked about these, the whale hip bones. This was a red circle in all of our notes in the last set. Leg bones and snakes. Once in a while, some snakes, like boa constrictors, I believe, will be born with little tiny limb buds. And then, of course, eyes left over, they don't need eyes in a cave. So you often will get blind organisms, blind fish, blind salamanders, millipedes, and lobsters, or crayfish. All right, number 5, biochemical evidence. So what does this tell us about evolution? So this is what they do nowadays. They compare DNA and amino acid sequences of organisms to see how well related they are. So when they discover a new species, they're walking in the Amazon and they discover a new monkey or primate or flower or insect, or if they're down deep in the ocean, and we've only explored about 10% of the ocean floor. They find almost new species every time they go down there, and they will collect one of the species or take a sample, and when they do, they will compare its DNA. Years ago, they didn't have this. Years ago, they made a lot of mistakes. Oh, this is related to that. Like the hyena, for example. You look at a hyena from Africa, and they look like a dog. Sort of, a weird dog, but they look like a dog. But they're not related to dogs. Once DNA and amino acid sequencing became available, they realized when they tested it that it's not related to a dog at all. It's related more to a weasel, to the weasel family. How crazy is that? So they, like I said, reveal new evolutionary relationships that we didn't know existed just by looking at the bones. And unfortunately, we can't go back in time and collect DNA from everything that's extinct. Can't do that. Now, there are very rare occasions where there's like maybe a woolly mammoth that's frozen in ice and gets defrosted, or a saber-toothed cat. Those kinds of things we can extract DNA from. But the whole Jurassic Park theory was that you can take blood stored in the abdomen of a biting mosquito and get dinosaur DNA from that, from 65 million years ago. But we know in reality that DNA, like many molecules, organic molecules, breaks down over time. And even if there may be pieces, you're not going to have a full set of DNA. So if organisms have similar DNA and RNA and amino acid sequences and similar proteins, scientists can infer, that's making an inference, that the organisms are related and share a common ancestor. So if you look at this table here, this is how much percent of amino acids that are identical to other species. We have 95% with a rhesus monkey. Monkeys have tails, apes don't. So if you see a chimpanzee or a gorilla, don't call it a monkey, please. Mice, we share 87%, 69% with chickens, 54% with frogs, and 14% with lampreys, which are a weird kind of fish that doesn't have a jaw. Jawless fish. So DNA in other forms. We're at the bottom of page 9, if you didn't know that. We're still moving on there. Of biochemical analysis were not used to study until the 1990s. So like I said, with the whole hyena thing, it was not known for many years that a hyena wasn't a dog. This technology did not exist during Darwin's time, obviously. So it's been added to the theory of evolution and provides the strongest support. Remember, theory, the word theory. When you see that word, theory, no matter what it's, Big Bang theory, theory of evolution, any theory, scientific theory, please know it's based on facts. And when new facts did not exist, okay? When things are added to a theory, the theory changes. Doesn't make it less strong, or it just changes it to be a new meaning, okay? So they look for genes on enzymes, they look for the amino acid sequences, common DNA between the organisms. And the more common they are, the more they're related. So these are different, this human and different apes that exist out there, like gorilla, that's pretty obvious, that's the gorilla. One of these is a chimpanzee. I don't know the exact scientific name of a chimpanzee. A bonobo, which looks like a chimpanzee, but it's different. So these are different kind of apes. And I bet you one of them is a Orangutan, too. Okay. I think that one actually might be orangutan. Pan troglodytes, but I don't know for sure. Anyway, so scientific names, and this is showing you all of the DNA sequences and how related they are, or the gene, sequences. And so compared to humans, chimps have 99.5% the same, and even a fruit fly, 25% with plants. 25% of our genes are shared with plants. How crazy is that? All right, so here's a question. Top of page 10, okay? I think. What would you suppose the closest living relative is to that animal? That little animal is called a shrew. I had one run up my arm once when I was gardening in New York. I was just pulling weeds from a rose garden bed when I was probably about your age. And this little thing that was kind of like a mouse, it ran up my arm and jumped off my shoulder. I was like, what? And it was a shrew. Maybe not that species, but the ones that live in North America. Smallest rodent in the world, smallest mammal in the world, shrew. But look at it. Doesn't look like, what, a rat? What is it? Here's another drawing of it, sticking its tongue out. Well, that one's a photograph, then a drawing. You might be surprised to find out that through DNA analysis, and this is not on your paper, I don't think, an elephant is its closest relative. Maybe some of you saw that little trunk-like thing and put 2 and 2 together, but still, an elephant, virtually the largest mammal, land mammal, and the smallest land mammal are related. What? It's crazy. All right, so now we're at the top of page 10, slide 69, and we're almost done with this set of notes because we have to stop to do an activity based upon this set. All right, so although natural selection may take many thousands of years to produce a change in a population, speciation, right? There have been some excellent examples of natural selection that have allowed scientists to study evolution in action. So not like if I had a time machine and I went back 10,000 years and then 10,000 years and then 10,000 years, that would be easy. Imagine the data that we could collect and all the holes in our theory that we could fill in. But we can't do that. So what we can do is look at evolution that occurs fast, like right in front of our eyes, over a certain number of small number of years that humans can witness it. And so we're gonna talk about changes in moth populations. And the first thing we're going to talk about is something called industrial melanism. Industrial melanism. So a long time ago, back in the Industrial Revolution, people started burning fossil fuels. Coal, specifically, for heat and power. Coal to heat their homes in the winter and to cook, and coal to boil water, which made steam, which powered wheels, steam-powered locomotives, steam-powered cars, steam-powered machinery for factories. And that made a lot of pollution, because when you burn coal and you don't filter out the smoke, which they didn't do, this black plume of smoke was coming out of everybody's chimneys. Can you imagine how much pollution that created in the environment? And it covered everything. Covered the buildings, covered the people, covered the streets, covered the trees. So now that you know what happened to change the environment, let's move on with the notes. Okay, so the appearance of dark colored insects, and these are called, we're going to refer to them as peppered moths because they look like they have pepper sprinkled on them, okay? In England, that blend against backgrounds darkened by industrial soot. And soot is that dust we talked about from burning coal. It looks like pencil graphite. When you rub it and you get it on your finger, that's what soot looks like. It's almost exactly looking like that. Pencil graphite's a little shinier because it's carbon and clay mixed together. There's no lead in your pencil, I hope you know that. There hasn't been lead in pencils for a very long time because lead is toxic. Imagine all the children in school writing with toxic instruments. Don't call it lead, it's graphite. Okay. And so what happened is that you could see there's a moth here, but it's hard to see because it's camouflaged on the light tree. Here's a light moth, here's a dark moth. Light, dark, hard to see. And this is what they look like, dark peppered moth on a light tree. So you've got the different variations and different colorations here in your mind. This is, normally, this moth would not be seen by a bird. It'd be hard for a bird to see that moth unless it was moving. So the moth is very good at camouflaging. But then comes the Industrial Revolution and soot, changing the trees to this color. Now which moth stands out? Which variation of the same species, light colored and dark colored, they're both the same species of moth, they're not different species, just a color variation, like we have gray squirrels and black squirrels. They're all the same species, just variations, okay? So now the tree's black, which makes the white moth stick out more. So what's the birds going to eat? The birds are going to eat the white moth and leave the dark moth. And so you're going to be left over with dark moths. So the selecting agent is the loss of camouflage in colored moths. And that results in more light-colored moths being eaten by birds. But then, towards the end of the Industrial Revolution, people started realizing how much damage they were doing to themselves and to their environment with burning so much coal. So then the invention of electricity, and so people started heating their homes differently. And they also had cleaner burning systems so that all that black soot wasn't coming out everywhere. And so then the black trees turned back into light trees again, and then it went back. The birds started eating the dark ones again. More of the population was now dark because the dark ones could hide, and then the light ones started coming back again. The frequency of the alleles in the population shifted. All right, let me just see something here. I think we are stopping somewhere soon. Oh, yeah, we're good. So the first, not the first, what we're going to be doing tomorrow is has all to do about that industrial melanism. There's an online simulation of this, okay, which you're going to need your laptops for. So make sure that you have your laptops tomorrow charged, and we're going to answer some questions, read a little bit online. It's all online. Do a little simulation, and then we're going to do an actual simulation, kind of arts and craftsy thing that we're going to do, and it's a competition between the other biology class. So you two are going to be competing to see something. I'll explain it tomorrow, okay? All right, so bacterial resistance to antibiotics. This is another way that we can see evolution happening right in front of us, okay? So new strains of bacteria. You may have heard of staph bacteria or staphylococcus bacteria, a staph infection. There is antibiotic resistant staph bacteria that even in the hospitals, they can't get rid of it. It's like there in the environment. You know, they clean those surgery rooms, the surgical rooms, as well as they can with antibiotic materials, but over time, the population of bacteria will develop a resistance to that antibiotic. So let's say I came in and I wiped the table down with a wipe. These were actually all wiped down last week by some students that came in and volunteered, but you should be wiping down your table when you come in. That's why the wipes are in here. But anyway, you wipe the table down and you kill 99.9% of the bacteria on your table. Why? Even when you look at a Lysol can, a Lysol spray, or when you look at the outside of the hand sanitizer, it says kills 99% of most illness-causing germs. 99.99. Well, if there are a billion bacteria on your seat from the last person who sat there, and you clean your seat, 0.001% of those bacteria, that's a lot of bacteria because they are resistant to it. They have something in them that's resisting dying. They have some thicker cell wall or something like that. And so they're going to reproduce and not die and pass their offspring on, you see? So that's what this means. So only bacteria who have a random variation to survive in that negative environment caused by hand sanitizer or antibiotics due to mutations, and that's going to make them resistant to that attack. And they'll survive and pass on those genes to their offspring. That's what bacteria antibiotic resistance means. Resistance to antibiotics means. So yeah, so you have MRSA, the MRSA, staph, smallpox, streptococcus, strep throat, E. coli. E. coli is Escherichia coli. That's the bacteria that's in your feces, your poop. Now, if you don't wash your hands very well after you go to the bathroom and use proper sanitation, you will, and then you come into the bathroom or come into the classroom, or someone else came into the classroom before you and sat down, and now you are eating in the classroom, which many of you do. You are essentially putting their poop in your mouth, because you touched the same things they did and you didn't clean it off first. That's why we always tell you that. That's why we always tell you to wash your hands. People think it's a joke, people laugh at it, but look around. So E. coli is the bacteria that's in feces. All right, so the main idea is this. And this isn't on your notes, just. So you have this organism that produces an enzyme, and then you have a mutant that produces no enzyme. You put the antibiotic on it, the normal one dies. The mutant, even though it's 0.001% of them out of a billion, that's like, a lot. So survives and continues to reproduce, and all of them are resistant to your attack. So it's like a war. And you've got 100 soldiers all coming in, and they're all wearing armor, but you've got armor-piercing bullets. They're all gone. 2 of them had extra armor, double layer of armor on. or a different kind of armor on that didn't allow the armor-piercing bullet to go through. So now those two remain alive to do whatever they're going to do. It's the same concept. Okay, insect resistance to insecticides. Same exact process. We see this happening in our lifetime. So appearance of new strains of insects, roaches, mosquitoes, flies that are resistant to pesticides like DDT. We don't use DDT anymore, but we did see this back in the day. We use other pesticides now. So the selecting agent in this case is not coloration, it's insecticide use. And only insects with the mutation which codes for not being killed by a chemical can survive and reproduce. So the first generation, you put pesticide on them. Most of them die. The red one gets left over. So look at the population percentage of red versus white. It's like 1 out of 10, right? Now it's 1/3 of them are red. So when they reproduce, that red gene is going to have a more chance of being in the gamete pool, in the gene pool, right? So the new application is sprayed, and now the population is almost all red. And they're resistant to the pesticide. Okay, we're almost done. Important facts. The environmental factor did not cause resistance. The resistance existed in the gene pool due to variations. So the soot did not cause the change. It was the variation in the gene pool, the white and black moths, that enabled the species to change, okay? It didn't cause it. And in bacteria or in insects, the pesticide didn't cause the resistance. It was already there in the variation of genes of the insects. The environmental factor is the selecting agent. So it weeds out. It's not the cause of the resistance, it's the weeding out agent. All right, you guys are being selected against now. Something happened in the environment, a chemical, a cold snap, where all the iguanas fell out of the sky. Well, not out of the sky, out of the trees. A poison cloud, either man-made or from a volcano, a tsunami, whatever. Some environmental factor came and selected against all of you, but all of you were left over. Over time, the gene frequency, that's the gene pool, can change, and resistance can be more frequent or common. Organisms did not will themselves into getting these traits. It's like COVID. I'm just going to think really, really hard, and I'll be resistant to it. Nope, it's just some of the population who are resistant and some who aren't, and maybe most who aren't. And maybe 90% of us get COVID before this is all over, and 10% don't. Those are the ones who are just, luckily, it's not like the bubonic plague, which we have a cure for now, but back then we didn't. Something like that comes in and just kills 90%. Imagine 90% of the people in this room just gone, and just you left over. That's what we're talking about. Okay, so now to test yourself, you are going to do, these are slides, the next up to slide 77. And there's a word bank on page 10, and it goes to the middle of the next page, page 11. So you're going to work on these right now for the remainder of the period with the people who are you sitting next to. Do not move your seats. And you have the rest of the class period to get that part done. And tomorrow we're going to do some moth creation. Industrial melanism practice.